I'd like to begin by thanking A4M, the American Academy for Anti-Aging Medicine, for their interest in sponsoring today's talk titled Cannabinoids and Adaptive Immunomodulation. And here are my attestations before we begin. I have backgrounds in biochemistry and agricultural science, and my biased perspectives derive from that of a career anesthesiologist trained in pain management and addiction medicine. My practical understanding of patient care is not that of a researcher per se, but likely consistent with that of a clinical pharmacist and clinical physiologist. Appreciating the complexity and integration of our human immune system's protective role in sustaining health is a timely subject, particularly now as a viral pandemic is challenging our clinical acumen and demanding our optimal understanding of effective treatment, safety, and efficacy. The two learning objective goals for this next half hour's talk are, first, and what I regard as the more critical objective, is to appreciate the unique mechanism of action of endocannabinoid receptors and their natural ligand's role in modulating physiology. And then our second objective will be to compare marijuana's primary bioactive chemicals, THC and CBD, for their potential impact on the immune system's integrated functions in fighting viruses. Primum non nocere, the Latin for first do no harm, is a guiding maxim for clinicians, and concern for balancing risk with benefit is central to treatment decisions in patient care. Most responsible clinicians will not advise their patients to pursue a particular treatment regimen without conclusive clinical evidence and peer review confirmation regarding the relative safety and efficacy of a given treatment. Yet the compassionate physician will also be mindful of the ethical obligation to ease suffering with whatever tools might be available and wherever possible. In this context, the medicinal use of cannabis currently presents a conundrum in need of bioethical rationale. The complexity of the evolving legal landscape for cannabinoid medicines will continue to require our attention, and physicians should act accordingly by first acknowledging that the treatment of intolerable symptoms to provide humane relief is a noble task that society bestows upon its healers. Well documented for over 5,000 years as a botanical medicine impacting both the mind and body, the discovery of endogenous cannabinoid receptors coupled with evidence-based reports indicating therapeutic efficacy from cannabis has renewed considerations for its therapeutic use. Cannabis sativa preparations were used for centuries in Asian medicine to reduce the severity of pain, inflammation, and asthma. Its therapeutic acceptance as a medicine evolved globally well into the 20th century, such that most major pharmaceutical companies included cannabis preparations within their formularies. This presentation emphasizes how appreciating the biologic context of how and why endogenous cannabinoid receptors function will allow healthcare providers to better interpret limited evidence-based data and preclinical trials. Among the receptor systems identified in the last century, several have important implications for treatment, such as the dopamine and serotonin systems. But none hold quite the significance of the endogenous cannabinoid system as it plays a regulatory role with all other bodily systems, including our immune system, to maintain certain critical physiologic balances sometimes referred to as homeostasis. Cannabinoid receptors are part of the Class A family of G-protein-coupled receptors, which are implicated in nearly all physiological functions of the peripheral and central nervous systems and in various peripheral organs. The endocannabinoid receptor system fulfills an essential regulatory role in the body's biochemistry and physiology, operating to maintain physiological system balances by keeping their functions within tenable parameters. Many different types of receptors can influence the activity of one another by enhancing or inhibiting their effects. But the endocannabinoid receptor system attenuates the action of all other receptor systems, including those of the endorphin receptor system central to stress and pain responses. Cannabinoid receptors are so ubiquitous and important that a 2013 National Institutes of Health extensive review concluded that modulating endocannabinoid system activity may have therapeutic potential in almost all diseases affecting humans. In summary, the endocannabinoid receptor system is the central regulatory system responsible for maintaining all life-sustaining balances with nature, not only in human beings as recently evolved higher mammals, but also in all animals, dating back to even include invertebrates that evolved 500 million years ago. It is noteworthy that another life form family, insects, have evolved different strategies to successfully survive. By the mid-1990s, in large part due to the study of the cannabis plant's bioactive chemicals, a previously unknown lipid-based receptor system capable of impacting all aspects of human physiology, including our immune responses to viral pathogens, was revealed. For all of the importance of plant-human interactions in identifying endogenous mechanisms and targets for analgesia, future therapeutic applications will have less to do with the plant's chemicals and more to do with other pharmacological ways to manipulate the endocannabinoid receptor system. This integrating system appears to be the body's principal regulatory mechanism, including in the brain, where it is known to affect memory, pain perception, and mood, among other things. All physiological systems' functions are subordinate to endocannabinoid receptor systems' effects, and the abundance of cannabinoid receptors in the human body is most remarkable, with roughly 4 times more than dopamine D2 receptors and 12 times more than mu-opiate receptors. The endocannabinoid receptor system differs from other receptor systems not just by being the only lipid-based neurotransmitter system. It is also unique in that it has not one but 2 ligands that exert their collective effects on 2 types of lipophilic cannabinoid receptors designated as CB1 and CB2. By comparison, the dopaminergic system has a single ligand neurotransmitter, dopamine, that is capable of acting on 5 different receptors, while the endorphins are known to act on at least 20 different receptors. The endocannabinoid receptor system also employs a unique retrograde feedback loop to affect presynaptic signaling. Both of the endogenous cannabinoids, 2-arachidonyl glycerol, abbreviated as 2AG, and N-arachidonyl ethanolamine, also known as anandamide, and dubbed anandamide from the Sanskrit word for bliss, describing its balancing impact, are found on the postsynaptic neuron membrane within a lipid ring encircling synapses that keep the endocannabinoids and their receptors sequestered from their aqueous surroundings. They are produced on-site and on demand. These cannabinoids then travel retrograde, backwards, to the presynaptic cannabinoid receptor associated with that transmitting neuron to modulate the release of all neurotransmitters. Endocannabinoid receptor activation allows it to operate as a constant fine-tuning master system, influencing all other neurotransmitters released to keep physiological systems in balance with life stressors. These receptors' partial or total activation can exert both positive and negative impact on the release of any and all neurons' neurotransmitters to constantly keep their physiologic effects in balance with life's changing demands. The components of the endocannabinoid receptor system are expressed in nearly all parts of the human body throughout every stage of life, and they exert influence on nearly all other receptor systems' neurotransmitters, including dopamine, serotonin, melatonin, and acetylcholine. The integrated systemic functions of endocannabinoids also scientifically describe the holistic and yet seemingly elusive brain-gut connection's dynamic interface with the microbiome. A brief review of what we know about endocannabinoid receptors' clinical impact helps understand the healing potential of endocannabinoid receptors, particularly their impact on the brain and nervous system. Due to extensive study of the cannabis plant's primary psychoactive chemical, delta-9 tetrahydrocannabinol, or THC, much is known about CB1 receptors that are primarily found in the central nervous system. These CB1 receptors' effects on the brain are known in part through researching THC's effects to clinically impact how we eat, sleep, relax, and even how we selectively forget. In contrast, CB2 receptors are located throughout the body and are there to protect us from the consequences of injury and disease. The essential pivotal balances provided through CB2 receptors are those that sustain life, and lack of functional endocannabinoid or immune systems will rapidly advance to death. Upon conception and continuing until death, the actions of messages that are produced by endocannabinoids, alone or in combination with other mediators, support all basic life-sustaining functions. From the perspective that all animals' genetic ability to evolve relies on maintaining an effective balance with life's continuous stressors, a healthy animal's naturalized response requires an effective endocannabinoid system's nurturing to ensure its ultimate survival. The immune system is known to play key roles in detecting malfunctioning cells inside our bodies, and through processes such as apoptosis or cell death, it ensures that these cells do not continue to grow and become tumors. The ability to kill cells is a crucial element of a healthy functional immune system that is responsible for maintaining delicate homeostatic balances between growth and decline. If, for example, there is too much cell death, autoimmune diseases can result, while too little cell death can create favorable environments for malignancies. In ancient times, practitioners of traditional Chinese medicine and acupuncture conceptually surmised that longevity derived from life-sustaining energy called qi that flows through the abdomen rather than through the brain, heart, or lungs. Research by Ahmad et al. In 2013 used serial PET scans to confirm that CB2 endocannabinoid receptors are primarily located in the abdomen, suggesting that longevity and health does indeed appear to be coordinated through the gut, where most of these receptors are located and function to orchestrate the immune system's diversified roles in maintaining health. The discovery of CB1 and CB2 cannabinoid receptors, as well as their endogenous ligands, renewed interest in fatty acid amides as modulators of immune responses. Endocannabinoid receptors' natural ligands, anandamide and 2-AG, are known to also be synthesized by cells derived from immune lineage, and it is clear that more research is needed to understand the intrinsic role endocannabinoids play in the immune system's healthy functional response to pathogens. Of particular interest, cannabinoid receptors signaling in response to injury by disease can confer therapeutic effects by downregulating inflammatory cytokine expression. In 2005, Klein summarized that a wealth of information indicates that cannabis-derived drugs have immunosuppressive and anti-inflammatory properties. On the basis of their mode of action, the therapeutic usefulness of these drugs in an optimal immune system's response to chronic inflammatory diseases is now being reassessed. Certain cannabinoids may bring potential therapeutic benefits because they can inhibit the production of pro-inflammatory cytokines triggered by COVID-19's caustic spike glycoproteins. In 2000, Berdyashev reviewed cannabinoid receptors and their regulation of immune responses. Cannabinoid research was noted as developing in two major directions: concerning the neurobehavioral properties of cannabinoids, primarily CB1 effects, and concerning the impact of cannabinoids on disease and the immune system, primarily CB2 effects. So even 20 years ago, it seemed likely that endocannabinoid signaling played an important attenuating role in the vigorous onset of immune responses, but the exact role for endocannabinoids and their receptors in the maintenance of immune system homeostasis, or the development of immune system disorders, is just now beginning to be understood. The effects of cannabis plant-derived chemicals on brain function has been extensively studied over the past 50 years and are well appreciated for their consequential impact to public health. These considerations are in large part responsible for marijuana's continuing status as a Schedule I prohibited substance. More recently, the immunoregulatory role of cannabinoids responding to infectious diseases has been summarized by Hernandez Cervantes et al. In 2017, to conclude that the administration of cannabinoids during critical stages of perinatal development suggests that prenatal cannabis exposure leads to a less efficient immune system function in adulthood, potentially increasing the risk of infectious diseases. While THC has generally been regarded as leading to unfavorable effects on several parameters impacting an ideal balanced immune response, our developing understanding of the physiologic challenges resulting from the novel coronavirus includes exaggerated cytokine and inflammatory processes in certain patients for which plant derived exogenous cannabinoids, particularly CBD or cannabidiol, appear to have therapeutic potential. Delta tetrahydrocannabinol's effects on several other well known viral pathogen models have been studied. THC could be harmful for a host infected with the influenza virus because THC administration appears to diminish the immune response against influenza. In the case of these viral infections, viral particle contact with cannabinoid compounds can negatively modulate some immune responses parameters. During human immunodeficiency virus infection, there is a reduction in CD4 plus or T helper cell lymphocytes and interferon gamma concentrations. During simian immunodeficiency virus infection, the activity of natural killer cells is inhibited, resulting in increased host mortality, and as with influenza virus infections, the viral load is increased. In vitro studies showed that hepatitis C virus and herpes simplex virus replication is increased in response to several plant cannabinoids, and in contrast, the use of certain synthetic cannabinoid agents during an infection with Thieler's virus, an experimental model for multiple sclerosis study, indicated that they may be good therapeutic targets for treating multiple sclerosis due to the fact that they promote remyelination. CB2 receptor functions play a particularly important protective role by mitigating the physiologic consequences of disease. CB2 receptors are generally thought of as being plentiful in the abdomen, in proximity to where the majority of cells of immune lineage are known to exist, but are cellularly ubiquitous in that they have been identified not only in vascular cells, but also in adipocytes, B lymphocytes, and macrophages. CB2 receptors are primarily associated with cells of innate immune lineage derived from the bone marrow. In the central nervous system, CB2 receptor expression is associated with responses to injury by modulating inflammation. CB2 receptors help maintain pivotal balances that are able to respond when and where an organism is threatened by the effects of disease or injury. In a clinical sense, corrective physiologic changes modulated through CB2 receptors may be those which allow a disease to become chronic, when an inability to respond to the progression of disease over time would otherwise result in death. This possibility emphasizes the clinical importance of future innovations that exploit this receptor system's subtle yet essential health function. Attention has refocused on CB2 receptors not only because of their expression primarily in cells and tissues of the immune system, but also because more is being learned about their intricate involvement in immune function, and because their activation in animals is largely devoid of the psychotropic effects attributable to CB1 receptors' impacts on the brain. The emerging scientific data show that distinct plant-based phytocannabinoids can activate immune cells by receptor-mediated as well as by non-receptor-mediated influences that further complicate our current detailed understanding. A new wave of research and mounting anecdotal evidence point towards cannabinoids having an adaptive immunomodulating effect rather than just simply suppressing immune activity, and that the bioactive chemicals in cannabis potentially interact with our immune system by impacting its complex strategic balances. Stimulation of our CB2 receptors by endogenous cannabinoids are known to suppress inflammation. The interactions between cytokine proteins that regulate inflammatory responses, immune cells, and the endogenous cannabinoid system play important roles in mitigating neuroinflammation and neurodegeneration that appear to be emerging as post-acute sequelae of COVID-19. Cannabidiol, or CBD, is a negative allosteric modulator of CB1 receptors, meaning that it modifies some but not all of a cannabinoid receptor's normal functions. For instance, even though CBD does not activate CB1 receptors directly, It can slow the rapid degradation of the endocannabinoid anandamide to prolong and enhance its natural effects on CB1 receptors. CBD has shown promise as an alternative therapy for the clinical management of anxiety disorders based on its anxiolytic and antidepressant properties. As an allosteric modulator of endocannabinoid receptors, CBD is capable of attenuating some of the dysphoric side effects from THC, such as paranoia. Patients recovering from COVID-19 may experience situational stress triggered by residual chronic inflammation and autoimmune reactions. CBD appears useful to allay anxieties impacting the behavioral health of patients who may not only suffer physically, but also psychologically or even spiritually from stress and COVID-19 recovery-related concerns. In 2015, Cabral et al. Reported that certain plant-derived and synthetic cannabinoids, as well as endocannabinoids, can modulate immune system functions. This immunomodulatory role of endocannabinoids suggests that 2-AG may be the more physiologically relevant endocannabinoid for CB2 receptors, playing the leading role in maintaining an overall balance of the immune system's function within a host. Because CB2 receptor activation's clinical implications dampen inflammatory responses in the absence of psychotropic effects on the brain, exploiting these particular receptors as molecular targets to attenuate inflammation linked to pathogenic disorders such as multiple sclerosis seems promising. Phytocannabinoids, such as CBD, have many targets other than CB1 and CB2 receptors, and CBD may be considered as the epitome of polypharmacy. It can interface with a host of other enzymes, transporters, ion channels, ligand-gated ion channels, GPCRs, and nuclear hormone receptors, and CBD can stimulate adenosine receptors and provide indirect pain relief by modulating other receptors indirectly. Perhaps most significant among these for CBD may be the transient receptor potential (TRP) channels, particularly those designated as transient receptor potential vanilloid 1 (TRPV1) channels, which appear to contribute to the control of inflammation, pain, and temperature sensation. Both CBD and anandamide can stimulate TRPV1 channel activity. The extent to which CBD appears to activate and over time may desensitize TRPV1 through receptor downregulation helps explain CBD's analgesic and anti-inflammatory effects. Other minor phytocannabinoids, but not THC, also appear active on these TRPV1 receptors. The complexity of endocannabinoid receptor system function, coupled with CBD's potential for polypharmacy, creates difficulties with the translational application of in vitro studies of cannabinoids' effects on the immune system's function, with those of in vivo responses extending similar effects to infectious disease processes. Whether or not this might influence a given patient's response to the novel coronavirus is poorly understood. However, it appears clear that the recent increase in marijuana's recreational use has revealed its combustion smoke's deleterious effects on pulmonary defense mechanisms against bacteria and viral infections. 2019 also saw life-threatening pulmonary complications subsequent to the increased use of vaporizing devices to deliver cannabis oil concentrates. While infectious diseases do not appear to have been responsible, black market contaminants added to inhaled cannabis oil concentrates progressed to significant reactive airway pneumonitis. The Centers for Disease Control urged doctors to report suspected cases to their state health agencies, and the Food and Drug Administration is collecting information about illnesses related to e-cigarettes and vaping. Because patients with COVID-19 can exhibit significant respiratory compromise, alternative methods to deliver medical cannabis's bioactive chemicals are currently undergoing greater consideration. The ability of cannabinoids to both suppress and enhance components of immune function lends credence to the notion that the endocannabinoid system is involved in bidirectional immunomodulation to maintain physiologic balances, keeping inflammation in check under healthy conditions, but enabling an inflammatory response when needed to fight infections. In their 2020 summary of the immune responses regulated by cannabidiol, Nichols and Kaplan concluded that current data overwhelmingly support the notion that CBD is immune suppressive and that its mechanisms involve the induction of apoptosis and promotion of regulatory cells, which in turn influence control over other immune cell targets, and through direct suppression, attenuating the activation of several immune cell types. Collectively, research findings support the additional investigation of cannabinoids in general. However, the landmark National Academies of Sciences, Engineering, and Medicine report emphasized that many of the studies in which the effects of whole plant cannabis on the immune system were evaluated possess significant shortcomings in experimental design, such as small numbers of study participants, and that this precludes drawing conclusions concerning the effects of cannabis on immune competence in humans with any reasonable level of certainty. Today in 2020, we're confronting an ironically myopic vision of the cannabis plant's limited, but nevertheless high-quality peer-reviewed evidence-based data concerning cannabidiol. CBD has an impressive safety profile with its best evidence for treating intractable epilepsy, but it also looks promising for treating schizophrenia, anxiety, and inflammatory disorders. CBD's dosing should be carefully integrated as part of a patient's overall pharmacological management because it can potentially induce cytochrome P450 enzymes, suggesting that it may have potential clinical relevance concerning the breakdown of other medications. Despite barriers to research, substantial evidence of the potential of certain cannabinoids is evolving and is consistent with what is known about the purpose of the endocannabinoid receptor system. Nevertheless, discussions regarding CBD's efficacy at this point should be considered as anecdotal reports lacking significant support of evidence-based data. When considering CBD's recent rescheduling to Schedule 5, now permitting its legal access, there may be a timely opportunity to responsibly research its clinical potential to mitigate COVID-19 related inflammation on considerably larger study samples. Particularly at the lower dose ranges of CBD reported to help treat anxiety, and due to a lack of significant side effects, CBD could sensibly be considered as a plausible option to add as an adjunct treatment along with plasma therapy, remdesivir, or with other new antivirals used to treat SARS-CoV-2-induced lung and systemic inflammation. To wrap things up from where we started, first, do no harm is attributed to the Greek physician Hippocrates, who responsibly documented and built upon his findings to historically be regarded as the father of medicine. The moral imperative of first do no harm is erroneously attributed to the Hippocratic oath of medicine conferred upon new doctors commencing with their noble careers as physician healers. However, first do no harm in fact derives from another of Hippocrates' writings titled Of the Epidemics, and it is apparent that these bioethical considerations are particularly relevant right now during a global pandemic healthcare crisis. The reality is that when difficult, urgent decisions must be made during a pandemic, it's hard to apply a first do no harm dictum because estimates of risk and benefit are notably uncertain and prone to error. Nevertheless, it is a reminder that we need high-quality research to help better understand the consequential balance of risks and benefits for whichever tests and treatments we recommend. Ultimately, it is also a reminder that doctors should neither overestimate their capacity to heal nor underestimate their capacity to cause harm. Clinicians suddenly faced with so many patients in need of care have obligations to not just feel compassion, but to do what is possible to alleviate suffering without causing harm. The wide search for safe and effective solutions to coronavirus has rightfully included medicines derived from cannabis. Perhaps most significant with traditional understandings of health is that Hippocrates is also credited with having said that nature itself is the best physician. As clinicians consider how to apply new discoveries, they may do well to heed the ethical standards which physicians have historically promised to uphold. whether the oath of Maimonides to seek the strength, time, and opportunity to correct acquired beliefs and extend the domain of knowledge, or that of the Hippocratic Oath to apply for the benefit of the sick all measures that are required.